Why lipids are biomicromolecules but found in the acid insoluble pool. Lipids are usually present in your membranes, like the cell membrane, which is made of phospholipid, bilayer of phospholipid. So these membranes, when they are broken by trichloroacetic acid, they are not broken fully. They are broken into pieces which form vesicles. Like they are broken like this. You see the bilayer, and they form vesicles. These vesicles do not dissolve in acid. Hence, they come under acid insoluble pool. But they are not true macromolecules, biomacromolecules, because their molecular weight is less than, it is 18 to 800 Dalton. Then we have this chemical analysis of living tissue. This is basically for knowing the organic compounds present in a living tissue. Ash analysis, whereas, is for knowing which inorganic ions or inorganic compounds are present. So you take your living tissue, anything, piece of liver or a vegetable, and that constitutes your wet weight. Then you're drying it, which constitutes your dry weight. H2 is removed, dehydration. Then you're performing combustion, that is heating in the presence of oxygen, and you are removing all the, let's say carbon is present, you're combusting, I guess. So then it releases as CO2, water as H2 vapors, NH3, etc. And now it forms ash. So this, hence called ash analysis. Ash contains inorganic compounds like SO4, 2 sulfate, phosphate, calcium, magnesium, chlorine, potassium, sodium, NaCl, CaCO3, etc., which we saw in the previous box. Most abundant intracellular ion inside the cell is K+, potassium. Most abundant extracellular ion is Na+, sodium. These both are important to remember. Okay. Metabolites of biomolecules. You have two kinds of metabolites, your primary and secondary. Primary metabolites are universal, present in all living organisms. They have a known function. They are basically what all you learned: lipids, proteins, nucleic acids, carbohydrates. Secondary metabolites, they occur in plants, fungi, and microbes. They do not have a known function. Not all of them, all of the functions are known to us. Many of them have use in human welfare and ecological importance, some of them. Example: pigments, drugs, lecithins, alkaloids, terpenoids, toxins, essential oils, polymeric substances. We have a table which is important. Okay. This you can remember with this mnemonic. Deepika Padukone is late for premium. D is your drugs. P is polymeric substances, if I'm not wrong. Okay. LATE. Sorry, P is for pigments. L is for lecithins, A is for alkaloids, T is terpenoids, C is essential oils, and T is also for toxins. P is polymeric substances. Just look at this table. Pigments are carotenoids and anthocyanins. Alkaloids are morphine, codeine, etc. Terpenoids, they have terpenes. You see monoterpenes, diterpenes. Essential oils are lemongrass oil. Toxins, abrin, ricin, lecithins. Lectins. Okay, lectins. Lectins. Concavalin A, concanavalin. Drugs, vinblastine, curcumin, etc. Polymeric substances, rubber, gum, cellulose. These polymers, cellulose is polymer of beta glucose, and these others. Okay. And then we have amino acids. So amino acids are biomicromolecules, building blocks of proteins, less than 1000 Daltons. Filtrate, they come under the filtrate acid soluble pool. These are basically substituted methane. So you have your methane, CHHHH. Now in these three of them, we are substituting it with different groups. That is your carboxyl group, amine group, and a variable R functional group. Your H is the same. So amino acid, basically amino, your NH2, and acid, COH. This is a variable group, and H. And the chemical and physical properties of amino acids come from carboxyl group, amino group, and your variable component, functional R group. And now NH2 and COH are found attached to the same carbon. This carbon is called alpha carbon, and this acid is called alpha amino acid. That's what is written here. And there are different types of amino acids based on different R groups. Only 20 types of amino acids form proteins. Now these three are very important. Remember as GAS, gas. Glycine. Now substitute R with H. That's the simplest amino acid you can do. When you're keeping H, it's glycine. CH3, alanine. CH2H, serine. Now other important things. There are aromatic amino acids. When R group is an aromatic ring, benzene or its derivatives. There are tyrosine, tryptophan, phenylalanine. Sulfur containing amino acids, methionine and cysteine. Neutral amino acids, where you have COH and NH2, like basically 1. This is actually equal to 1. You have like these, basically glycine is alanine, serine, even valine. Acidic amino acids are where in place of R you have another component that has COH, or basically COH. So these two examples are aspartic acid or aspartate, glutamic acid or glutamate. And basic amino acid, same. In place of R, you're substituting NH2 or something related to NH2. Like let's say CH2, NH2. You're substituting that. So lysine, arginine, and histidine. Very important are lysine and arginine, which you find in basic non-histone proteins in animal cells and all, and in histone proteins in prokaryotes. So they are basic, right? They are lysine and arginine. And amino acids are divided into two categories based on the formation in our body or not. Non-essential and essential. Non-essential are formed in body. They are not essential in diet. They don't have to be supplemented through diet. Whereas essential are not formed in body. Did I say not formed or formed? Okay. Essential in diet. Remember this with this mnemonic. Try this VIP mol. Where try tryptophan. This is threonine. His is histidine. V is valine. I is isoleucine. P is phenylalanine. M is methionine. A is arginine. L is lysine. L is leucine. I hope you can see. Okay, due to the ionizable nature of NH2 and COH group, structure of amino acids changes in different pH solutions. So just look at this. Here, let's say it's an acidic pH, less acidic condition. Your H plus is going off from COH. So this forms like, this is more acidic, of course. CO minus. Sometimes let's say this H is going to this one. It is CO minus and this is NH3 plus. This is called Zwitterion basically. This is important. This is dipolar. It has two poles. Like polar, like two charges, Zwitterion. And Zwitterion and NH3 plus. Your COH is there. It's gaining a H plus from the whichever solution it's kept in. Then you have your proteins, which are made from of course your amino acids. Biomacromolecules, acid insoluble pool, retentate more than 10 Dalton, linear chain of amino acids linked by peptide bond. Okay, so here if your amino acids, okay, this one thing, always heteropolymer of amino acids. So you can never find a protein that has only histidine or only lysine or only glycine. Never. There's always a glycine or histidine or something. It's always a heteropolymer of amino acids. How peptide bond is formed. So you have your, let's say R1 group, some amino acid and one more amino acid. These three components are the same. NH2, COH. Now OH from here and H from this are removed. They are removed, and this forms dehydration. This forms, you have your O. I'll just draw here, I guess. 1 second. You have your let's say H2N. This COH you basically know, it's like this O connected to OH. Now from here it forms, you have your NH2 Cr1 H C, this O is there, C double bond O. Now for this you are having your N, H2 is gone, so you are having your N which has H and then your C same. Okay, now in this you see this is one protein. Let's say it's basically polypeptide, but it's a protein. This bond, this is your peptide bond. Basically C double bond O N H or CO NH. And in this, if you see, you have to the left side, this is your left, this is your right. Your left you have your N terminal, and to the right you have your C terminal. Remember this, this is important. To the left side you'll have your N terminal, to the right side you'll always have your C terminal. First carbons has the left N terminal, last carbon has the C terminal. So basically that's what is written here. Peptide bond COH of one amino acid reacts with NH2 of second amino acid by removal of H2. Okay, this is how you indicate removal of H2 when this bond is formed. And like that you have your R3, R4, R5, many amino acids forming. First amino acid, it has N terminal, left end. Last amino acid, C terminal, right end. Structure of proteins. The primary structure is the linear sequence of amino acids. Basically what you see here, what you see here, what you see here, it's linear. This is the, it has only peptide bond, so it's very unstable. Secondary structure is the alpha helix or your beta plated. Both of them are secondary structures. They have peptide bond plus hydrogen bonding. and they're always right-handed helices or helices. And this basically your N-terminal, C-terminal, always right hand. And these are also, let's say they're pleated like this. They have hydrogen bond between two helices or between two other peptide bonds. These are pleated, let's say pleats. Okay, this one 3-degree amino acids. It's basically like this, you see. Let's see, these you have some sides here, like you can see some hollow woolen kind of wool thing, you see, hollow woolen ball kind of thing. They are active sites. These are your some of these hollow things are your active sites for substrates to act on. I'll just write substrates, subs here. And just remember this, tertiary, it's absolutely necessary for many biological activities like enzymes, enzymatic reactions. Okay, they are long chain of polypeptides folding upon itself like a hollow woolen ball. That's it. And yeah, of course, somewhere it's starting, somewhere you have your N-terminal, somewhere ending your C-terminal. Now, the 4-degree quaternary structure is basically you have disulfide bonds between the 3-degree structures, you can say. The disulfide bonds actually give the quaternary structure of protein. And one of these examples is insulin. And okay, insulin, just protein's example is insulin, which has alpha and beta secondary structures. It has 21 alpha amino acids and 30 beta amino acids. So 51. It's not important this one, but hemoglobin, this is a PYQ, 2 alpha and 2 beta. So it has 4 subunits. I'll just write here if required. It has 4 subunits. That is 2 alpha and 2 beta. It's the most abundant protein in whole biosphere is rubisco. I can tell the full form in a second. It's ribulose biphosphate carboxylase oxygenase. Okay, and in animal kingdom it's collagen. It's intercellular ground substance. I can just show it in this box in NCERT. Okay, this is what you should see. Collagen, it's intercellular ground substance between the cells and connective tissue we find. Trypsin, it's an enzyme. Insulin, it's a hormone. Antibody fights infectious agents. We know it's protein, specifically glycoprotein, but yeah. Receptors, some of the receptors. And GLUT4, that is glucose transport. GLU, glucose T transporter, enables glucose transport into cells. It's a PYQ. So yeah, I think that much. Okay, now we are going to lipids. Lipids are water insoluble, but they are, and also acid insoluble fraction, but they are soluble in some organic compounds like acetone, benzene, and all those. There are some simple lipids are fatty acid. Fatty acid is basically RCOH. This is the basic formula, where this R can be methyl 2 as much as 19 carbons. So R can be C1, C2 to 19 carbons, whereas fatty acid will be from 1 to 20 carbons, like C2 to 20, if you understood. I hope so. Like the minimum number of carbons in a fatty acid is 2, the maximum is of course 20. In these fatty acids, there are 2 types, saturated, unsaturated. Saturated are the ones that do not have any double bonds. Example, palmitic acid and stearic acid. Just remember, like, you catch the steering with palm, like palms is steering. So palm has 16 carbons. You see, when you're forming, it's usually CH3, CH2, 18, like that, if it's a 19 carbon chain. CH3, COH. Here your 2 carbons are done, so here you get 14, 16, 18, so you're 16 in stearic acid. In unsaturated fatty acids, you find double bonds. If they have one double bond, mono unsaturated. More than one double bond, poly unsaturated fatty acids, MUFA, PUFA, whatever. Mono unsaturated fatty acid is oleic acid. It has 18 carbons. Polyunsaturated is linoleic acid, 18. Linolenic acid, 18. Arachidonic acid, 20. What you should remember from these is palmitic acid has 16 carbons, stearic acid has 18 carbons, arachidonic acid has 20 carbons. Very important. There in NCERT, previous question. So yeah. These 3 are not important. I guess they'll be used in some other chapters. So yeah, others all, oleic acid, linoleic acid and linolenic acid have 18 carbons. Now, one more simple lipid is glycerol. It's basically trihydroxypropane, or if you want to be like propane 1,3, 2,3, triol, OH is all suffix. Many lipids, these are your simple lipids, but many lipids have both fatty acids, your fatty acids RCOH, and glycerol. So how is the bond formed? This is just your ester bond. You have your CH2, CH2, OH, and this is, let's say, R1 something, and COH. OH from COH and H from here are removed. Let's say if monoglyceride, like glyceride is usually what is glycerol plus fatty acid. Glyceride, it's monoglyceride. 2 are gone, diglyceride. 3 are the triglyceride. So we are removing 3 molecules of water. We get 3 ester bonds. This is your triglyceride, which is, in heart health, cardiovascular health, you use triglycerides and low density lipoprotein, cholesterol, high density lipoprotein, HDL cholesterol and all. Okay, fine, whatever. We have digressed. Glycerol is esterified. Okay, this one you remember, very important. 1 glycerol with 3 fatty acids forms triglyceride. You have 1 glyceride and 3 fatty acids for 1 triglyceride. Glycerol is esterified with fatty acid to form mono, di or triglyceride. And ester bond is your COOH, whatever. Lipids or triglycerides are of 2 types. Again, saturated, unsaturated. Saturated lipids, saturated fatty acids, they have high melting point. So that usually solid in room temperature. Ghee, butter. Unsaturated are liquid in room temperature. Oil, or just I'm writing gingerly oil. This is a question from fingertips. So yeah. Now, lipids we usually know in membranes, we find it as phospholipid. Especially plasma membrane, we know it's a bilayer of phospholipid. So, what is a phospholipid basically? It has lipid plus phosphorylated organic compound or phosphate basically. It occurs in cell membrane and also neural tissue with complex lipids. They didn't mention, but you find that lipids in the myelin sheath, which is made of phospholipid, which is called sphingomyelin. Most common phospholipid is lecithin. This is the structure of lecithin. You can see here there's R1, there's some R2, and then here a phosphorylated compound is present. Like, okay, I guess, yeah. This is basically our phosphate, like, phosphate, and yeah, another compound. Then cholesterol, it's a derived lipid. It has no fatty acids and no ester bonds. It has 17 carbons, 4 rings, and OH group is present, and it's like cholesterol. Just see the structure of cholesterol, which is very important. I hope it's there somewhere. Yeah, this is cholesterol. Okay, let's just see this page. What we know: lecithin, you have your CH, this one, this is R group, this is some phosphate one, this is your cholesterol, this is triglyceride, you see R1, R2, R3, glycerol, dihydroxypropane, fatty acid, this is palmitic acid, 14. If there are 16, 18 carbons totally, stearic acid. If there's 18 here and this, it's arachidonic acid. That's it from here till now. And we know glycine, alanine, and serine. If it's H, if it's CH3, CH2OH. This is hydroxymethyl, this is methyl, this is just hydro, whatever. Carbohydrates basic unit is monosaccharide. Monosaccharide is a basic unit of carbohydrate. The generally it has this formula CnH2nOn. Let's just say glucose, C6H12O6. It's a hexose sugar, like 6 carbon sugar. Ribose is a pentose sugar, 5 carbon, 5 C5H10O5. Fructose has the same formula as glucose, but the position of OH group, or they both are like isomers basically. Their position, it's actually okay. Glucose is a aldose sugar. Fructose is a ketose sugar. I hope I wrote it somewhere right. Okay, yeah, I wrote here. Glucose is aldose. Okay, so glucose is aldose, fructose is ketose sugar. Ribose, see, from ribose, when you're removing one oxygen, deoxy, this becomes a deoxyribose. C5H10, C5H10, O5, O4. This is used in our DNA, this is in RNA. Oligosaccharide. So this is one monosaccharide, one ribose or one pentose or one fructose or one glucose. Oligosaccharides are 2 to 9 monosaccharides, like 2 units of glucose or 1 unit of glucose and fructose, 1 unit of glucose and fructose and something like that. Okay, 2 to 9 units of monosaccharides that are formed with glycosidic bond, which is of course dehydration. Again, I'll explain how to form glycosidic bond. Disaccharides, 2 means disaccharides. We have 3 main examples, very important. Maltose is glucose plus glucose. Lactose is glucose plus galactose, and sucrose is glucose plus fructose. That's and lactose is milk sugar, sucrose is cane sugar. It's commercial or table sugar that we actually have. This transport sugar in phloem. This is very important again. Polysaccharides are more or equal to 10 monosaccharides. Again, glycosidic bond. There are 2 types again in this. Homopolysaccharides, which have the same units. Starch has alpha glucose as the same unit, like it's a homopolymer of alpha glucose. Glycogen is again glycogen is again alpha glucose. This is basically in plant, this is in humans, animal kingdom. Cellulose is beta glucose, chitin is NAG, you'll know. Inulin is a polymer of fructose. Very important. Inulin is a polymer of fructose. Fructose.